Thank you. And I'd like to thank the MPOC and in particular Dr. Sundrum for the invitation to share with you some of my research this morning. And I'll be talking about palm tocotrienol and how it attenuates stroke injury by improving collateral circulation during cerebral ischemia. Stroke worldwide is a large problem. More than 15 million stroke events occur each year. And associated with stroke injury is irreversible damage to the brain, paralysis on one or both sides of the body, difficulty with walking, eating, or other daily activities, and loss of ability to speak or understand speech. According to the National Stroke Association of Malaysia, there are 40,000 stroke events in Malaysia alone every year. And in Malaysia, it's the leading cause of long-term severe disability, and it's the third leading cause of death. Stroke comes in two varieties, as was mentioned by the previous speakers: hemorrhagic stroke, which is the rupture of a blood vessel in the brain, and ischemic stroke, which my lab focuses on, and this is caused by the blockage of a blood vessel, causing local ischemia and disruption of blood flow. And this accounts for the majority of all stroke events, roughly 87%. And in particular, my interest is in looking at how blood vessels of the brain respond to this ischemic stroke injury and attempt to compensate for this blockage in blood flow. If you look at how we study and how cerebral vascular anatomy is presented to students today, there are some very rudimentary, simplistic cartoons that are used to try to describe the very complex cerebral vascular anatomy. This is one example from a scientific medical textbook showing how these larger vessels, which traverse the leptomeningeal space, these larger arteries, which then branch off into smaller arterioles, penetrate into the brain to supply blood to regions of the brain. When you look at what we actually see in vivo, it's much, much more complex. This is an image generated by my lab. It's a corrosion cast. We do this by injecting a polymer resin into the blood vessels during stroke. In this case, the stroke-affected hemisphere being in this region here. You're looking at the top of the brain down. This is from a mouse. Mouse brain's about the size of the tip of my pinky finger. But what's remarkable here is you can appreciate the complexity of the vascular system in the brain. And in particular, I'm going to zoom in here on a portion of this. And you can see truly how magnificent the density of these blood vessels are. And it almost appears as if there's no room for any other cell type, like there's no room for neurons or glia to actually interact. But in between this mesh and this network of blood vessels, there are neurons and astrocytes and all different cell types which interact, each and every one, directly with the blood vessel. And it's this remarkable network of collaterals and blood vessels which supply blood to the brain and ultimately give brain tissue the oxygen and glucose that it needs to thrive and survive. I'll come back to this image later. I'd like to shift gears, though, quickly and talk about vitamin E. And a brief background is discovered in 1926 by Dr. Herbert Evans at the University of California, Berkeley. And at that time, it was discovered it was known as a fertility factor. He called it originally, actually, vitamin X, not vitamin E, but it was 10 years later when he published in the Journal of Biological Chemistry that he assigned a chemical name for it. At that time, alpha-tocopherol, tocospheros, the ability to bear offspring, comes from the Greek word tocopherol. And there he described the biological activities of vitamin E for the first time. Now today we know that there are 8 naturally occurring vitamin E families, broken down into 2 classes, tocopherols and tocotrienols. And this was covered previously, so I won't spend a lot of time here. What should be known, though, is the antioxidant activity of the molecules is ascribed to the chromanol head here. And the chromanol head of alpha-tocopherol is identical to that of alpha-tocotrienol. What confers unique biological activity, which is independent of antioxidant function, is actually in the phytyl tail, where we see those 3 unsaturations. Now, in the US, if I go to the pharmacy and buy vitamin E capsules off the shelf, I'm only getting one type of vitamin E, and that is alpha-tocopherol. Despite saying vitamin E on the label, the mixed tocotrienols are not contained here. And it should be noted that my vitamin E research is all focused on tocotrienols, in particular palm tocotrienols, and we use Tocovid Superbio as the source of these, generously provided by Hovid Industries. There's a growing body of literature, as Dr. Sen shared, on tocotrienols in the literature, and recent developments of antioxidant unique or independent activity. And as Dr. Sen shared, there's a large body of research on the unique neuroprotective properties of the vitamin E family members belonging to tocotrienol. And that's where I actually started my PhD work in his lab back in 2002. It was actually, I think, the second day in the lab that I began working on this project, which was published then in 2005 in the American Heart Association Journal Stroke. We saw this slide already this morning. I won't spend time here, but we did see that with the supplementation regimen that we used in this particular study, a 50 milligram dose of alpha tocotrienol, orally gavaged, it was delivered to brain tissue in significant quantity. Very low amount in our vitamin E deficient controls detected in brain. We're able to achieve roughly 1/4 the amount of alpha tocopherol level of alpha tocotrienol in the brain after our supplementation regimen. That small amount of alpha tocotrienol which was delivered to brain, however, was highly neuroprotective in the context of reducing the size of the stroke injury. This is actually not an MRI image, this is a TTC stain, this is a viability stain showing viability of mitochondria in the neural tissue. And you can see that there are loss of the mitochondrial viability, denoted by this pale region here. It's much less with tocotrienol supplementation. We quantified that and saw that the stroke lesion was significantly reduced with tocotrienol supplementation. And moving from here, Dr. Sen and our group has published a number of articles on the neuroprotective properties of natural vitamin E. And as mentioned in the previous lecture, neuroprotective agents, however, alone, while they've shown great promise in preclinical studies, have not translated to clinical efficacy yet. There have been more than 200 neuroprotective clinical trials which have all failed at this point in the clinic. And it's led to ask some whether neuroprotection is an attainable goal for stroke therapeutics. And so moving forward, it's become recognized, as Dr. Sen shared, that we need a multimodal approach for stroke therapy. With that in mind, I began a series of research with the support of Dr. Sen to study or to identify some of these multimodal mechanisms of how palm tocotrienols, as I'll abbreviate throughout this presentation, TE, how these protect against stroke. To do this, we wanted to develop a new model, a translational model of acute ischemic stroke. We recognized very early on some of the limitations of the small animal models. It's also shared by the previous speaker. We wanted to, before we went to clinical trials, use a large animal setting with a brain that closely mimicked that of the human. And so we also wanted a minimally invasive approach that enabled real-time monitoring of cerebral vascular blood flow in our model that we developed. We wanted an accurate and reproducible means of confirming the occlusion or stroke event. And we certainly needed desired anatomical and pathological relevance to the human brain and stroke condition. We first explored opportunities using non-human primates. In the US, there are certainly, and elsewhere in the world, certainly a number of ethical considerations with using non-human primates in research, and in particular also very high cost associated with that line of investigation. We next tried a porcine or a pig model of ischemic stroke. The pig cerebral vascular anatomy, however, has a very unique anatomical structure called the rete mirabile, which humans do not possess. And so when looking or trying to identify vascular protective effects, we didn't think this model was a good fit for our work. We finally went with the canine stroke model. Importantly, canines have a history of use to study hemorrhagic stroke already. And more importantly, I think they have a gray matter to white matter ratio that closely approximates humans. This graph I don't expect you to be able to see, but this is published in Proceedings of the National Academy of Science, showing the relationship between canine stroke brain and gray matter to white matter ratio and that of humans. They're very close to one another. More importantly, too, the canine brain has gyrencephalic undulations. These are the folds in the brain tissue, which are not present in the mouse brain. The mouse and rat brain have lysencephalic or smooth brain surfaces. And so again, anatomically very relevant model to use the canine, as it's very close to that of humans. And you saw this slide already too. I had the opportunity in my PhD years to be trained by an interventional neuroradiologist, Dr. Greg Christophorides, who at that time was at the University at Ohio State. He has since moved on to teach at University of Chicago in Illinois. But he equipped me with the tools necessary, the endovascular approach, to navigate blood vessels in the canine. We actually went through the femoral arteries, very far away from the brain, to minimize any inflammation or injury that would be associated with our endovascular approach. We then navigated, at this point you see I'm going around the heart with my catheters and micro wires on my way up to the brain, where we would cause the stroke event by occluding the middle cerebral artery. We also leaned on some of our strengths in MRI. This is actually a magnetic resonance angiography that allows us to look at the cerebrovascular anatomy non-invasively. We use this to design our approach. So we recognized very early on that the internal carotid arteries have this tortuous loop. It almost forms a figure 8 that you see here. And clinically, in humans, this would be the normal route to approach the middle cerebral artery, which you see coming off the circle of Willis here. Unfortunately, in the canine model, that wasn't able to be navigated endovascularly. So we developed a new approach where we actually went along the basilar artery, across the posterior communicating artery, and then to the MCA to cause the stroke event. There's a cartoon of our approach here. So here we're actually beginning from the vertebral arteries, which then combine to form the basilar on the way up to the circle of Willis. So we'd navigate endovascularly around the PCOM, or posterior communicating artery, and we deploy an embolic coil, an occlusive coil that would block the M1 segment of the middle cerebral artery. This is also clinically appropriate because 75% roughly of all strokes which are presented clinically are MCA territory infarcts. So to cause the stroke event, we used a very clinically relevant tool. This is called a Guglielmi detachable coil. Clinically, they would use these to fill an aneurysm, bulge in a blood vessel. Here we're using it to actually block the blood vessel itself. So these can be deployed from a microcatheter, and once deployed, you can see they take on this 3D shape, which can become occlusive. And so we would send a microwire to the origin of the MCA, or the M1 segment of the MCA. We could use contrast injection dyes to show that the MCA was perfusing normally before we would deploy the coil. We then deploy the coil across the M1 segment here. You can see the coil in place showing up on the X-ray here. We do another contrast injection, and now you can see the MCA is not filling as it did here. It's blocked. We can then go to the opposite side and show the MCA of the opposite hemisphere is filling normally. And we'd induce the stroke for 60 minutes and then pull the occluder back out so we can retrieve these coils to model reperfusion injury as well. And we can then do another contrast injection and show that both MCAs are filling normally, although you'll note that the stroke-affected one fills a little slower. And that is also seen in clinics following strokes in humans. So we published this model in 2008 in the Proceedings of the National Academy of Science, and this set the basis then for our preclinical trial to use this model to test tocotrienol, palm-derived tocotrienol vitamin E. So in our trial, we had 20 mongrel canines that had 10 weeks of prophylactic supplementation of a tocotrienol-enriched gel capsule. In this case, it was Hovid's 200 milligram Tocovid Super Bio. They received it after meals twice per day, or the control group was just a vitamin E deficient placebo control. They underwent 60 minutes of MCAO, or middle cerebral artery occlusion, followed by 6-hour and 24-hour magnetic resonance imaging. And this was the primary outcome from the study, in which we saw, as we did in our preclinical mouse models, robust protection with tocotrienol supplementation. Here's tocotrienol concentration in brain tissue. You can see we didn't detect any in our placebo control group. We had roughly 80 nanomoles per milligram protein tocotrienol following 10 weeks of prophylactic supplementation according to the regimen I just described. And that's roughly 1/10 of the amount of alpha tocopherol in the brain. But again, despite the lesser amount of alpha tocotrienol compared to tocopherol, robust protection. And this is at 6 hours following stroke. Normally after stroke, you have an evolution of the stroke lesion over time. So in the placebo group, you see the size of the stroke lesion grows from 6 hours to 24 hours. And remarkably, we didn't see any of that inflammation or growth associated with edema and swelling with our tocotrienol supplemented group, meaning that the size of the lesion stayed the same from 6 hours to 24 hours. These are representative coronal slices at the 6-hour time point from our placebo group and our tocotrienol supplemented group. Infarct showing up in the bright white colored lesion here. At 24 hours, we can get a much higher resolution image using T2 weighted imaging. And you can see, again, the large size of the stroke-induced lesion in the placebo group and the reduction that was afforded by tocotrienol supplementation prior to the stroke injury. Another tool we can use to look at this, to appreciate this, is 3D volumetric reconstruction. So we take all those slices, we put them together, we apply pseudo-color to highlight the difference between the stroke-affected tissue and the healthy tissue. This is a representative placebo canine, and a representative tocotrienol canine with infarct volumes that are very close to the average for both groups. This is at 24 hours after stroke, and we can now cut through the brain pixel by pixel, and you can appreciate the size of the stroke injury of the placebo-supplemented canine versus the tocotrienol supplemented. Look at white matter injury, very relevant, as again discussed by the last speaker. We can use diffusion tensor imaging, or DTI. This is an approach to look at the white matter fiber tracts themselves. This is, again, a 3D representation of those in a placebo and a tocotrienol supplemented canine. I'm going to go back and start this from the beginning again. And you can see the stroke injury in this hemisphere caused a significant loss of these white matter fiber tracts. And actually, because of the swelling and the edema associated with the injury, it also caused loss of the fiber tracts in the contralateral hemisphere. There was actually no stroke there, but because of the swelling and edema, it injured the opposite side of the brain as well. And here with tocotrienol supplementation, again, robust protection. You see protection in the corpus callosum as well. So the fiber tracts which connect one hemisphere to the other are also protected and very robust. Now getting to the multimodal mechanism of protection here, we began after we had the primary lesion volume data returned to us. We unblinded ourselves from the study at that time and began to do a retrospective review of the cerebral angiograms that we acquired during the surgeries themselves. I'd like to draw your attention to panel A here, and this is a tocotrienol supplement canine on the bottom and a placebo supplemented on top. And this is before we've occluded the vessel, so you can see the MCA is outlined by this green angle here in both cases. And this is the MCA territory, and the MCA is filling normally in the arterial phase here. Then we place our occluder across the M1 segment of the MCA, and now you can see it's not filling anymore in this region. But what we found remarkably when we looked at a contrast injection in the contralateral hemisphere, you can see the other MCA fills normally in both cases, but there are the presence of these collateral vessels. These are small arterioles which were supplying blood back to the otherwise stroke-affected MCA. I apologize that it's a little difficult to see here, the contrast. It shows up much better on my screen, and if you go to the publication that I'll show you the reference for here, you can appreciate it much better. But you can see there are actually much more collaterals in this representative tocotrienol supplemented brain, again allowing blood to flow from the contralateral hemisphere back to the stroke-affected MCA territory to supply blood to the otherwise occluded MCA. And fewer, far fewer, with our placebo canine. And we can use a clinical scale to score these. So clinicians know, neurologists know that some people, some humans have very good collaterals, some people have very poor collaterals. And they know that if you have good collaterals, you generally have better stroke outcomes than if you have poor. And so they've developed these scales to actually score collaterals in the brain. And we use that same scale to score our collaterals for our canines. And we saw that those that were supplemented with tocotrienol vitamin E had much better collateral circulation in the MCA territory that should be occluded than the placebos. And that correlated very nicely with the size of the stroke lesion, such that if you had very good collaterals, you had very small stroke-induced infarct volume. The black diamonds here being tocotrienol supplemented canines, the open diamonds being the placebos. And you can see most of the placebos had worse collateral scores and significantly larger stroke volumes. Getting to mechanism then, we used our strength in laser capture microdissection. So this is a technique that's relatively new, the ability to put slides, sections of tissue on a slide, and then actually using a laser to cut out just the collateral blood vessels at this point, collect those in a tube, do downstream gene protein expression analyses. This first panel is just showing that what I've captured is very specific for endothelial cells. We use VWF as an endothelial cell marker. It's very low expression of neuronal or astrocyte markers, glial markers. So again, I'm selecting very specifically the population of blood vessels from the stroke-affected brain that I believe to be collaterals. And then I began querying or screening gene expression of known arteriogenic factors, arteriogenesis being different from angiogenesis. Arteriogenesis is the remodeling of blood vessels in the brain to improve collateral circulation. Angiogenesis, most people are familiar with as new blood vessel growth. This is not new blood vessel growth that we're seeing. We're seeing remodeling of existing blood vessels that improve collateral flow. And we see that in increased expression of chloride intracellular channel 1, CLIC1, in the stroke-affected hemisphere of the tocotrienol supplemented brain, and also increased expression of TIMP1, tissue inhibitor of metalloproteinase 1, in tocotrienol supplemented canine in the stroke-affected hemisphere. Angiogenic marker, VEGF, no difference in expression. So again, supporting that this was an arteriogenic remodeling of collateral blood vessels in the brain and not an angiogenic effect. TIMP1 targets matrix metalloproteinase 2, or MMP2. And this slide is simply showing that there was no change in expression of MMP2 itself in our two supplementation groups, but activity of MMP2 was significantly inhibited because of higher TIMP1 expression in the brain. And so we published this in the Journal of Cerebral Blood Flow and Metabolism, 2011. This is Nature publication journal. And since then, I've gone on to ask some new molecular mechanistic questions. So coming back to my corrosion cast image here, all of this research now is funded by a 4-year grant from the American Heart Association to study mechanisms of how tocotrienol improves collateral blood flow to stroke-affected brain. What I didn't tell you previously is that this mouse brain is supplemented. This guy was supplemented with tocotrienol for 10 weeks prior to inducing stroke. And you see this blood vessel here in panel C, this is a collateral. And I've applied a pseudo color here, and you can see aspects of this collateral reaching out and reconnecting to distal aspects of the otherwise what should be occluded middle cerebral artery, which comes up here and branches up here. And it's these collateral blood vessels which allow blood from the contralateral hemisphere to come back through, supply these aspects of blood vessels in the leptomeningeal space, and ultimately retrogradely fill portions of the MCA to supply blood. And so using the mouse model, which has much better availability of molecular biology tools, and using our laser capture approach, we can again select for just these collaterals. Here I'm using an approach by labeling all of these collaterals with a FITC conjugated lectin. So I'm perfusing this during ischemia, so only the blood vessels that are perfused will have this green staining here. So you can see much more of these with tocotrienol supplementation than our placebo control. We can quantify that, so again, much better collateral blood flow with tocotrienol supplementation. We use CD31 as a marker for all endothelial cells. You see no change really in endothelial cell abundance, so it's not angiogenesis. These are not new blood vessels. These are remodeling of existing ones again. And those same arteriogenic markers we've identified also appear in our mouse models of stroke. And moving beyond this now, the importance of this is what does it mean to have a smaller stroke lesion? What does it mean to have reduction in stroke size by 50%, by 75%? What are the functional outcomes that change, and how could we eventually use this to improve the quality of life of stroke survivors? So we're using very sophisticated behavioral testing software that automates recording and collection of movement of the animals in an open field space. After stroke, these animals typically have hemiparesis or paralysis on one side of their bodies, so they can only ambulate in circles like this. With tocotrienol supplementation, we see that animals can ambulate much more in straight lines and explore the corners of their environment like they normally would. We can automatically quantify from the software. There's no manipulation by anyone in my lab to come up with these numbers. The software simply spits these out for us, which is nice. We can see that the tocotrienol supplemented mice after stroke walk farther, they spend more time being mobile, and they move faster than the placebo controls. Again, showing that there's a functional relevance to this protection that we're observing. I'd like to finish my talk now with a few closing thoughts, a little abstract here, but when I'm not in the lab and studying stroke biology, one of the hobbies of mine is actually reading and studying space exploration. And I'm a big fan, in particular, of studying the Apollo missions, which were the missions that undertook the taking of the first man to the moon. These missions are remarkable not only for the scientific achievement, of course, or the engineering achievement, the feat that came with this, which I would argue is the most exciting or the greatest scientific achievement of the last century. But there was also, if you work in scientific research, a lot of interest for me anyhow to look at how the logistics of this were handled. How did we actually achieve this monumental goal of putting a man on the moon? And if you go back to the history books and study it and look at it, it wasn't a scientist who got us there first. It was actually an administrator, a politician, in this case John F. Kennedy, who in 1961, on May 25th, said in 10 years, we are going to put a man on the moon. Now, unbeknownst to his chief scientific advisor, Chris Kraft, who would actually then be the director of these missions, Chris Kraft had no idea that John F. Kennedy was going to be making that statement, or making that audacious statement that in 10 years we'll put a man on the moon. And he's actually quoted as saying he thought he was crazy. There's no way we could achieve this. We couldn't put a man on the moon in 10 years. Well, it turns out they didn't take 10 years, they did it in 8. And the way that they did it is phenomenal. It took a team of 400,000 people, 400,000 individuals, and a unique blend of government officials and administrators, and it took a lot of people in academia and industry working together to achieve this, 400,000 people. And not everyone was an astronaut or even a scientist for that matter. The spacesuits which were used to go to the moon and back were actually hand-stitched by a seamstress at that time. And so Eleanor Foraker, who was the spacesuit seamstress, there are 11 layers to these spacesuits. She would hand-stitch each and every one of those. And I love this quote by her. When she saw the guys, the Apollo 11 mission, which the first men on the moon, jumping up and down, doing all this activity on the moon, she said she wasn't worried at all until she saw them jumping up and down and really stretching out those seams of their spacesuits. But anyhow, the point here I want to make is that it takes a large team with a lot of people with a lot of divergent, different talents to accomplish something like this. And so you look at the teams involved here. You have government units again, you have academic units, you have industry partners, all working together towards that common goal. And whether your achievement or your goal is to put a man on the moon or try to find a stroke therapy, it takes that same mix. And I'm very proud to be working with the team here of government officials and with industry partners and academic partners to achieve this mission. And moving forward, I guess I would like to challenge the group, as John F. Kennedy did so many years ago, to identify what that goal is for tocotrienol research. Help us form the best team that we can, with adequately resourced partners as well, to achieve that mission. I think we're certainly on our way there. Wrap up, I'd just like to thank my partners, certainly Dr. Sen and Dr. Khanna, who's to speak next, members of my lab team. And I'd like to leave a quote from Thomas Edison, native son of Ohio, the state that I'm from in the U.S., who said that surgery, diet, and antiseptics, these three are the vital things of the future for preserving the health of humanity. And there were never so many able, active minds at work on the problems of diseases as now, and all their discoveries are tending to the simple truth that you can't improve on nature. And certainly, I think when we get to spend some more time looking not only at the tocotrienol component of vitamin E and palm oil, I think we see more evidence from carotenoids and other types of these biological compounds coming out of palm oil. I think we'll see that this simple truth will continue to prove true. Thank you.